Okay, how are you today? Okay, this screencast, and we've already done this, this screencast is going to take us through the different phases of mitosis. All right, I wanted to point out to you that in your Cornell, you're going to need to highlight each one of these phases with a diagram and a drawing each for each one, and not the drawing you see on this slide right here, this little drawing that I made for you. This is textbook chapter 5, and chapter 5 handles this extremely well. So great pictures, great pictures. I'm just going to take you through the parts and all of the points that you need to know about the mitosis phase of the cell cycle. All right. So again, mitosis is known as the process of nuclear division. Okay, and that's where DNA is replicated, and the replicated molecules are arranged into two separate nuclei. So mitosis is always preceded by interphase, the cell cycle, and then has four distinct phases: prophase, metaphase, anaphase, and telophase. Now, the division of the cell, the actual dividing of the cell into two new cells, happens at the same time as the final stage. So even though we'll take cytokinesis separate, it happens at the final. It happens at the same time as telophase. All right. So what you see up on the board here is prophase. All right. Prophase is one. So the DNA has replicated. The DNA has, or say organelles have replicated as well, and now we're ready to divide. All right, so before mitosis, you have prophase. All right, actual first part there. So, and what happens here? All right, so what you see here, the chromosomes become visible. That process is called supercoiling. They supercoil together, and they condense. And by visible, we mean you can actually see them under the microscope. It's the only time in the cell cycle that you can actually see. That is during the M phase, during mitosis. All right, chromosomes become visible. The nuclear membrane breaks down. All right, so if I had a nuclear membrane here, which I don't, I do have one here, but I would put like little holes into it or spaces there. Nuclear membrane breaks down. The nucleolus breaks down. We need to get rid of the nucleus, basically, so it's the DNA. All right, the centromere attaches to each chromatid. All right, so this is the same piece of genetic information. This is the same piece of genetic information. Together, you have this chromosome number 1 to match up with another chromosome number 1. All right, so the pair centrosomes also begin to move to the opposite poles. So the centrioles move to the opposite poles as part of the centrosome region. Spindle fibers and asters begin to form. Again, none of that is depicted in this picture here. Do not use this picture for your Cornell. Do use either of the pictures on page 86, fully labeled. They're very nice. This picture gives you an idea, but it doesn't give you nearly enough information to write it. All right. Or you could use the pictures if you pause either of the two animations, one on the animal cell and one on the plant cell that we just put up yesterday. All right, another dog. Metaphase. So here's the classic metaphase picture: is lining up. Chromosomes line up, so the chromosomes move condensed and move towards the center, okay, of the cell and line up. So you're going to have lining up. All right, you're going to have spindle fibers developed, and you're going to have the spindle fibers attached to each chromatid. That's tid, I-T-I-D. All right, so the sisters have lined up, sister, 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 at the equator, and this is called an metaphase equator. It's sometimes called a metaphase plate as well. Okay, metaphase plate as well. That's a T. All right, chromatids are attached to the spindle. So we're going to get ready to pull them apart. All right. All right, so let me go over that again. So you have the spindle microtubules from both centrosomes, which are kind of here. Okay, connect at the centromere, which is keeping the chromatid together of each chromosome. So this is a chromosome containing two chromatid. All right. So the spindle fibers there have lengthened and the chromosomes align. They move towards the middle, towards the metaphase plate. Again, there's a good paragraph for each one of these on page 86. That is page 86, the pictures. Okay, so metaphase. Chromosomes meet in the middle. Anaphase, pull apart. The classic picture you see here that you will see on page 87. Okay, anaphase, little blurb, little figure headings. Use it. Use the book this time. So what's happening here? Let's bring up, and you got the dog going off. All right. So this is the separation of the chromatid. So the centromere cracks, breaks, and the chromatids begin to be pulled to either end of the cell. Actually, the pulling occurs because the protein gets shorter and shorter. So it's not an actual pulling like a tug of war. So anaphase pulls it apart. And right at the very end of that, even though not depicted here or in any picture, cytokinesis begins. So anaphase again, spindle fibers shorten and causes the chromatid to separate. Once they separate, they're each considered an individual chromosome again. So all of these are going to a new cell, and all of these are going to a new cell. And they need to be genetically identical, all right, move to the different parts of the cell. All right, pull apart. Now, the telophase picture often has and does have cytokinesis depicted as well, because you can't have one without the other. All right, so you see here the picture there, the unlabeled picture, and then you see the good telophase picture on page 87 in your textbook. And you got a picture of an animal cell and a plant cell on page 86 and 87 as well. So let's get this all up here. All right. Chromosomes reach the opposite ends of the cell. The nuclear membrane begins to reappear. The nucleolus begins to reappear inside. The spindle disappears. All right. The chromosomes unravel because they're going back to their interphase state. All right. And cytokinesis then is completed by the end of telophase. So again, cytokinesis is a different idea. It's the actual splitting of the cell. Prophase, metaphase, anaphase, telophase, nuclear division, the dividing of the nucleus into what's going to be. So this is the divided nucleus into two new cells. So we got the dog information. Now there are going to be two. All right, so information on page 86 and 87. Am I driving that home for you enough? Two animations to watch and a set of notes to form in your Cornells. Don't be limited to these 4 or 5 points. Enhance them and do give us fully labeled diagrams. Okay? This screencast is now done.